some of the tools that we're going to need to essentially deliberately genetically engineer cells and DNA are going to be DNA polymerases and DNA ligases derived from bacteria, and reverse transcriptase derived from retroviruses. This little arsenal of enzymes will allow us to artificially, in vitro, deliberately manipulate and successfully process different sources of DNA and engineer new combinations that have new, unique characteristics. Now, some of the first materials that we'll need to start with are pieces of DNA. We really can't use whole chromosomes. Those are much too large. So we're going to have to somehow make little bitty pieces or fragments. We can do this chemically with enzymes that we call restriction enzymes, or we could simply do it mechanically. And that was a technique that was used decades ago, is to simply take pieces of cells or pieces of nuclei and just put them in very high-speed blenders and chop them up into bits and pieces. But scientists have found in the intervening years that fragments of DNA produced by using restriction enzymes are much better tools. And so, how do we use those restriction enzymes, and what do we manufacture as a result? Well, we're going to create teeny little fragments that have sticky ends, pieces that we can splice together. Now we're going to find that if we take two sources of DNA, they could be two distinctly different sources of DNA, and we essentially nick or cut them using the same restriction enzyme, we're going to wind up with little tags or little pieces of sticky ends on the termini or the tip ends of all of the little pieces of DNA that essentially have the same complementary nucleotide sequences. That's why we call them sticky ends, is that they're tiny little pieces of single-stranded DNA sticking from the tail ends of fragments of DNA, and they can easily reanneal or hybridize or attach to small single-stranded pieces of complementary DNA sticking out of the tails of other fragments of DNA. When we assemble all these little fragments, just the little pieces, not having them attached to anything else, we call these collections of fragments of DNA that's been cut up with restriction endonucleases, a DNA library. And using these little mixtures, or using these DNA libraries, are successful ways that scientists have cut and pasted different sources or different fragments of DNA to get unique new combinations, or recombinant DNA. Once we've produced the restriction fragments, or these little pieces of DNA, double-stranded DNA with little sticky ends, there's all kinds of techniques and manipulations that we can perform on these fragments. We can essentially separate the fragments by their size or their mass in a process called electrophoresis. Once we've separated the fragments by their size and mass, then we can transfer those fragments from one matrix to another. It's called blotting. We can do all kinds of things once we've established and manufactured the fragments. But of course the trick is getting all the tools together, the enzymes and the building blocks and materials, to actually perform and make those fragments. And of course, one of the activities or things that we can do with many of these fragments is to make recombinant double-stranded DNA. Remember, recombinant double-stranded DNA, or recombinant DNA, or rDNA, are pieces of double-stranded DNA from two different sources that are spliced together. The This slide is an illustration of how restriction endonucleases can function. There are literally hundreds of different kinds of endonucleases that have been discovered. Endo means within, so these are enzymes, nucleases, and a nuclease is an enzyme that essentially has some kind of action on a nucleic acid. And any word that ends with ase automatically tells us it's an enzyme or biological catalyst. So an endonuclease is an enzyme that's going to catalyze the breaking, or what we call the cut or cleavage, of the phosphodiester bond along the outside of the DNA ladder between the two ends of whatever piece of DNA we're working on. So endo means within. We're cutting between the two ends of the double-stranded DNA, and the restriction endonucleases are catalyzing the breaking of the phosphodiester bond on both sides of the ladder. Notice the restriction site, the reason we call them restriction endonucleases, the restriction site is actually a unique sequence of nucleotides that the enzyme, the endonuclease, recognizes. It's essentially a binding site, or a direction, or almost like pointing an arrow to that piece of DNA that says or tells the enzyme, cut here. And that is the only place the enzyme will cut. And that's for lots of different types of endonucleases. The restriction site is different for each one. So it's like having a pair of chemical scissors that's looking and trying to find just a unique place on a piece of material or rope or whatever we're trying to cut. Well, that's what the restriction enzyme or restriction endonuclease is trying to do. Find that unique place being directed to a unique nucleotide sequence to essentially cut the sugar phosphate backbone. When we do that, or when the enzyme does that function, essentially on the DNA, it's going to weaken the hydrogen bonding between the two sides of the DNA ladder and eventually allow us to remove that fragment from the DNA ladder. Because if we weakened the phosphodiester bond or removed it, it's going to be much easier to separate the two sides of the ladder because the hydrogen bonds are not nearly that strong. So using different endonucleases, we can cut pieces of DNA into different sized fragments and then use those fragments to create recombinant DNA in a test tube or in a bacterium. This is a close-up image of how a restriction endonuclease might function and the production of sticky ends. Notice that on this little sequence of DNA, the restriction site, or the unique sequence of nucleotides, is illustrated. On the top, it's GAA-TTC. On the bottom, it's essentially an inverted repeat of the same sequence, just going in the opposite direction because of the anti-parallel construction of the DNA. But the restriction site is the same on both sides of the DNA ladder for this particular example restriction endonuclease. It is going to catalyze the cleavage of the phosphodiester bond between the first G and the A of this restriction site on both sides of the DNA ladder. What you have remaining are the two little tiny pieces of sticky ends, or two little pieces of single-stranded DNA on the tail end, well, not the tail end of the whole fragment, but on this little piece that's been produced on this DNA fragment. Notice that the cut site is, if we look at the overall whole structure of the DNA, is somewhere within the ladder, sort of somewhere inside or within this fragment. Here's a more specific example illustrating how we can use restriction endonucleases to create sticky ends on DNA from two different sources, and then splice those together to create recombinant DNA. In the illustration, we have a piece of DNA that's colored blue, representing chromosomal DNA, and a little ring of yellow DNA, which is representing plasmid DNA. If we use the same restriction endonuclease on all of these fragments or these pieces of DNA, they're all going to have the same sticky ends. If we allow the sticky ends to anneal, some of them are going to re-anneal to themselves, so the blues will go back together. Some of them, the yellows, will go back together. But occasionally, we will find that the DNA from the two different sources with the same sticky ends will anneal to each other and create our recombinant DNA, which is illustrated in the middle on the illustration on this slide. So now we have a piece of single circular DNA that's mostly yellow, but also has now this new blue piece of DNA spliced inside. This is exactly the kind of construct that we want, or the type of recombinant DNA that we want, to try and find unique sequences of DNA to produce the unique proteins that we might need, let's say, for the production of recombinant human insulin. Once we've manufactured our recombinant DNA, now we have to somehow transport it into our target cell. Now, the target cell initially, many years ago, of course, was simply transferring DNA from one bacterium to another, and particularly artificially creating these recombinant structures. So this little illustration simply shows how we could create the recombinant DNA and use the plasmid as the vector or the transport device to transfer genetic material from one bacterium to another, and hopefully with expression of those genes from the first bacterium to the second. Now, it was discovered that these kinds of techniques, recombinant DNA formation, the use of restriction endonucleases, all of these activities and mechanisms are naturally occurring processes in prokaryotes. But once scientists discovered that this naturally occurred, and that perhaps we could artificially perform similar kinds of tasks in the laboratory or in vitro with different sources of DNA, not just bacterial and viral DNA, we thought, well, gee, maybe we could do this with eukaryotic DNA too. Of course, that is one of the ultimate goals and objectives for most of the processes of molecular biotechnology and the history of molecular biotechnology. Ideally, we want to perhaps, let's say, cure a disease that's due to some type of genetic or gene malfunction by introducing a correct functional copy of the gene into the patient to cure their disease. That really was sort of the ultimate ideal many decades ago, and one of the fundamental reasons for the process and the formation and the encouragement in molecular biotechnology many years ago. Many years ago, for example, when we tried to treat diabetes, we had to use bovine insulin from cows or insulin from animals. And many of those animal proteins that are used to treat human diseases, some people will develop essentially an allergic reaction or some kind of immune response to that animal protein because it's not human protein or not closely related types of biochemicals. So the idea of manufacturing human proteins, like human insulin to treat diabetes, really was an admirable and ideal proposal. So when this kind of molecular biotechnology was discovered naturally occurring in bacteria, and then scientists determined that they could artificially perform the same function in the laboratory, it was thought, well, maybe we can do this with eukaryotic cells as well. And originally, they did try to use eukaryotic chromosomal DNA and the same kinds of enzyme tools, the restriction endonucleases and the DNA ligases, to create recombinant DNA fragments and then use vectors like plasmids and expression tools like bacteria to see if they could manufacture human products and human proteins. Originally, when they tried to use eukaryotic chromosomal DNA to create recombinant prokaryotic, eukaryotic pieces of DNA and manufacture human proteins, it just didn't work. Because human chromosomal or eukaryotic chromosomal DNA is not all transcribable and translatable. We found through the study and through the actual implementation of these molecular recombinant DNA technology tools and processes that eukaryotic chromosomal DNA is made up of what we call introns and exons. The introns are essentially regulatory binding sites. They have some other function other than to be transcribed and translated. Whereas the exons are the sequences of DNA that are supposed to be transcribed and translated. Well, the introns, in order for us to get a functional piece of DNA that we can actually eventually transcribe and translate, once we've made our recombinant DNA construct, the introns must be spliced out or somehow removed in order to make eventually a functional piece of DNA and then a functional piece of messenger RNA that can be translated. Kind of a tricky procedure. We're going to need something else to help us take RNA and make DNA. And up until the late 1970s and 1980s, this really was a stumbling block because nothing had been discovered that could allow us to essentially go backwards from RNA to DNA. The genetic dogma at the time was the flow of information was DNA to RNA to protein. How in the world are we going to get around this particular stumbling block? It was discovered that if we wanted to make human insulin, we had to go to the cells in the human pancreas that were manufacturing insulin and find the messenger RNA that was specifically used to manufacture insulin. And we needed to somehow make a DNA copy of that unique specific messenger RNA to get the exact nucleotide sequence of the DNA needed to manufacture insulin. Quite a problem. As it turns out, in the late 1970s, early 1980s, a unique enzyme was discovered in a group of viruses called retroviruses. Retroviruses are RNA viruses, but they are capable of producing DNA copies of the retroviral genome and splicing or inserting the DNA copy of the retroviral genome into the eukaryotic chromosomes with the formation of a provirus. So, the enzyme responsible for the synthesis of DNA from RNA, discovered in retroviruses, is reverse transcriptase. Reverse transcriptase then allows us to make essentially cDNA or copy DNA or complementary DNA copies of any kind of RNA. Doesn't matter what source, the enzyme will function or perform its function of making DNA copies of RNA. The RNA can be from any source. And so, sure enough, if scientists find the cells within the pancreas that are producing insulin, isolate RNA from those cells, and make copies or cDNA copies of that RNA, now we're going to have the unique DNA sequence free of introns that can be used and spliced into a vector, like a plasmid. Here we have a close-up image of using reverse transcriptase to make a DNA copy of RNA. And of course, ideally, we want to make a copy of mRNA to make a functional gene or a piece or sequence of nucleotides of DNA that can be transcribed and translated. We call those copies cDNA. The C can stand for copy, that's the usual assignment, or it could even be complementary. But in any case, once we've made lots and lots of copies, or cDNAs, we can also use those to form our DNA library. Regardless of how we generate the fragments, whether we mechanically cut the DNA up or simply use restriction enzymes with sticky ends that can be used, our cDNA library can be then spliced into some type of vector like a plasmid and then introduced into various types of cells, either prokaryotic or even eukaryotic cells. Of course, the original studies were all done in prokaryotes and then eventually perfected and applied in eukaryotic cell systems like yeast.